you know, obviously I'm Eddie Koo, and it's a great honor privilege to introduce Dr. Susan Taylor, our speaker today, and she's truly one of our trophy faculties at UCSD. She's widely acknowledged as a pioneer and really a world expert on the biochemistry of protein kinases, especially the cyclic AMP-dependent protein kinase A, PKA. She's born and raised in Wisconsin and attended University of Wisconsin-Madison for her undergraduate degree. She obtained her PhD from Johns Hopkins University, followed by a postdoc at the MRC Laboratory of Molecular Biology at University of Cambridge in the UK. She came to UCSD in 1972 and started her pioneering work on PKA and has been at UCSD ever since then, while launching her really amazing trajectory to academic stardom. She's now professor of chemistry and biochemistry, and also in the Department of Pharmacology at UCSD. Her awards are really too numerous to mention here, but included election to the National Academy of Sciences and the Institute of Medicine, and as a Howard Hughes Medical Institute investigator. 25 years ago, when I first came to UCSD, I got to know Palmer Taylor because of his work on the nicotinic acetylcholine receptor. And he frequently attended neurology grand rounds and also gave grand rounds. And now it's Susan's turn to begin some work where they have potential mechanistic links to AD, PD, and mitochondrial function. So we very much look forward to what I understand is her new work in the retina and how it might be relevant to neurodegenerative diseases. So please join me in welcoming Susan to the Shiley-Marcos ADRC Neurodegeneration Seminar. Thanks very much, Susan.
Oh, you're welcome, Eddie. And thank you for inviting me. For me, it's a real opportunity to talk to this group because I really would like to develop more collaborations. And even at this stage of my career, which, as you know, it's been a long time, I'm still learning. And you guys are the ones who are really teaching me so many new things now. So I'm going to go back to some of the history just because I think it's important. And the kinases, the first kinase was phosphorylase kinase, and then PKA a decade later in 1968 was the second protein kinase to be discovered. And so it's an old kinase. It's one of the very early ones. And this was all about glycogen metabolism. And then up at the top was Tony Hunter and the tyrosine kinases. That was a decade later, but I'm going to focus on PKA. And I'm also going to try to emphasize to you how kinases, how do they influence so many diseases? So many diseases are caused by mutations in kinases. Cancer, for sure, we hear all about, but cardiovascular disease, diabetes, and neurodegenerative diseases. Kinases are switches, and that they get tuned a little bit too high or a little bit too low, they cause a disease. So they're very much drivers of disease. And okay. All right, and so it was also, PKA was the first kinase to be sequenced. And so it was sequenced in 1981, and SARC was cloned in 1979, and it was thought to be a kinase, but nobody knew what a kinase looked like. So it wasn't until PKA was sequenced by the group in Seattle that Margaret Dehaas, she was a really early pioneer, recognized the homologies. And it's not super homologous, but she recognized these conserved motifs. And we had done a lot of work with labeling a particular lysine we knew was important at the active site. And so this is enough. It was enough for Tony Hunter and Steve Hanks to take 25 sequences and align them and make that basic kinome tree. It was now a big tree that included cancer biology people as well as all the metabolism people. And these are kind of the subdomains of that kinase core that's conserved. For me, stone that, is the chemical description of the molecule. And we wanted to know, what is this one lysine residue doing? And so my mantra from coming from Cambridge was always, you had to have a structure if you really wanted to understand function. And so that was 30 years ago, that first structure was solved. So I was still just an associate professor. And so, and it was really a major conceptual breakthrough, I think, for understanding what this kinase looked like in 3 dimensions. And we've learned in the ensuing all those 30 years, it's a very dynamic molecule. We didn't appreciate that from the first structure, but it's very dynamic. It's a switch. It gets turned on and off, just like the GTPases. And so what did we learn from that first structure? So this is our first kinase structure. And you learned the conserved fold. You knew all those kinases on that tree were going to have the same fold. Those motifs, the little white dots, this is our lysine, which was right at the active site as we have predicted. You learned how ATP binding, this is actually a novel ATP binding site that hadn't been described before. Peptide binding, you have this high affinity peptide binding. You kind of had everything captured here. And it's also an active kinase. And we know now most structures that we have are inactive kinases and they have disordered parts, but this was active. It was fully phosphorylated. And so it was really a template for the protein kinase family. And so I wanted to tell you not about the switch mechanism, but I wanted to tell you about this core. So this is just the kinase core, the N lobe and the C lobe that's conserved in all of those kinases. And this is PKA and PKC. And we've been doing a lot of work recently with Alexandra Newton. And you can model the core of any kinase based on the PKA structure. What does the active kinase look like? You can get a good model. But it's not active, just that core by itself. It really needs these tails. And PKA it has tails, an N-terminal tail and a C-terminal tail that wrap around it, and they regulate it, and it's not active without those tails. So I'm just going to tell you, here you can see in a movie how those tails wrap around the 2 cores, and they regulate it. And most kinases have other domains that are attached to it, and they like EGF receptor, PKC, and they regulate it. But PKA is relatively simple because it has these tails on there. And so I'm going to tell you about the AGC kinases. So PKC, PKG, the ROCK kinase, the GRKs, they all belong to this AGC subfamily. And they all have this conserved C-terminal tail. It's absolutely conserved in that whole family. And this is just superimposing a bunch of structures. We have lots of structures now. And if you kind of melt away the core of the kinase, you can see how that tail is really conserved and how this part gets organized. It's phosphorylated by mTOR, PKC, alpha, PDK1. It's highly regulated how you organize this tail, but the tail itself is conserved in all of those AGC kinases. So I wanted to just pause here in the middle of my little PKA story to tell you 2 things that I think I hope will be interesting to you and important as a way of thinking together about these molecules. And this is a new grant that just got funded by Alexandra and me. We're co-PIs on this, and it's about PKC gamma. And what Alexandra's found that if you look at PKC alpha, you find inactivating mutations are drivers of cancer, whereas activating mutations in PKC gamma drive ataxia and Alzheimer's disease. And so this grant is on the PKC gamma. And this concept is a really important one because it's just a little out of whack, a little too active, you get the ataxia. A little less active, you get cancer. So the isoforms are really important also. That's a main message. These isoforms are really, really important and they work differently. And so just to show you the mutations in the PKC gamma that drive ataxia, you can see they're scattered kind of all over those domains, but they're mostly in this C1 beta domain. And I kind of consider these disease mutations to be like a treasure chest of that you can find within there, maybe one that stabilizes as a state that is otherwise too dynamic to see. And so this was the basis for the grant that we're just starting to look at now. And hopefully we'll get a whole structure by cryo-EM of the kinase domain, as well as the C2, which is the calcium binding domain, and the C1 beta, which is the this is the calcium, this is the pH binding domain. And so, but you can see how the mutations cluster in this C1b domain in particular. And so can we model that? Can we use the kinase as a model and build a model of what the holoenzyme structure will look like, and then can we validate it? So that's one project that we're working on that I think is relevant for you as a group. The other that I was going to talk about entirely, but I'm just do a little bit here, is the LRRK2 structure, which again is a large multi-domain protein, over 2,500 amino acids, and there's a kinase domain here. And so how do LRRK2 mutations, you can see there are a few mutations here, a single mutation in this very large protein makes it a risk factor for Parkinson's disease. Why is that so? And how is LRRK2 regulated by phosphorylation and by this kinase domain? And so I'm going to tell you this because it's, I think, important for what I'd like to do now with Alzheimer's disease. And so this is LRRK2, and it shows at the bottom the phosphosites, actually 3 of those critical ones, this one, and this one, and one other one are PKA sites. And so PKA is involved in this as well, but you have the PD mutations that can create a protein that now becomes a risk factor for Parkinson's disease. And then you have the phosphorylation sites. So how can we dissect those and use our knowledge of kinases? And of course, here's the kinase domain, which is really a driver of all the conformational dynamics. And so what I did, this was in 2016, I put together this team that was funded by Michael J. Fox. And the 3 of us at UCSD, myself and the Andreas Lechner had just come, and Elizabeth Villa had recently come. And they do electron microscopy and electron tomography. And none of the 3 of us had worked on LRRK2 before at all. And so I said, we need to have a structure. There was no structure available. Could we pool our expertise and really begin to understand this? And so this consortium, which turned out to be really successful. Last summer, we published 2 papers. Andres' lab published a paper of the C-terminal RCKW domain at a 3.5 angstrom resolution. So this is really a wonderful model for drug discovery, showing how the kinase drives the conformation of this core. But Elizabeth did something that was even more amazing. She saw the structure of this active dimer. This is an inactive monomer. This is an active dimer, and it's wrapped around microtubules. And that's in cells, not purified. That's in cells. And I wanted just to tell you a little bit, because this was work that was done by Mark Ellison's lab in a number of years earlier. And they showed that like 3 of the 4 common LRRK2 mutants, Parkinson's disease mutants, and unlike wild type LRRK2, form these filaments. And so those filaments seem to be microtubules. Wild type didn't do it. But now we've been working with Daniela Boassa in the NICMER, and you can see as soon as you add a high affinity kinase inhibitor to wild type LRRK2, it forms these filaments in real time in cells, and it's reversible. If you wash away the inhibitor, it's reversible. This is what I want to do with looking at some of the filaments that are associated with Alzheimer's disease. I want to do this live cell imaging. That's a really important goal, I think, for Alzheimer's disease. This is a really nice tool. And how do these kinase and phosphatase inhibitors alter docking? And so we're now doing a project with Michael J. Fox, with Alexandra as well. Alexandra is an expert on phosphatases as well as kinases, and the kinases and the phosphatases work together. But I'll just show you this structure. This is a structure that Elizabeth solved. This is now in whole cells. Tomography, you just make a thin layer. And she could show that the LRRK2 bound as polymers around microtubules. And you can see on the right, this is the dimer, as I told you. It's an active dimer that forms polymers that wrap around microtubules. And it's really extraordinary. Elizabeth is a world expert in this. It really goes beyond cryo-EM, and it takes the molecule within the cell. You don't have to purify it. And this is a mutant. This is one of those mutants of LRRK2 that spontaneously docks onto microtubules. So this is just another example of how those disease mutations can be a real treasure chest for trapping something that otherwise is too dynamic to see. And we did a lot of work in this time on the kinase domain and showing how the kinase domain actually drives the dynamic changes in LRRK2. And we're trying to do now the same thing with PKA. How can PKA drive some of these neurodegenerative diseases in a similar way using similar tools? And this is just showing now in Andres' structure how the kinase domain, here's the N lobe and here's the C lobe, and there's two really, really strong helices that wrap themselves around the kinase domain, and there are these key phosphorylation sites. So how do they control the dynamics? And most importantly, how can we identify inhibitor sites that are targets for drug discovery that are not just the active site of the kinase? There's just many sites here that are really going to be, I think, great allosteric sites. That's my little bit about LRRK2 and what we're doing there. But now I want to come back to PKA and talk about the holoenzyme. And Shmuel was one of my really, really close friends from Israel who thought as much as I do about PKA and the mechanisms that control its function. And he sent me this. It's a stone. It's a small stone. It's on my desk in my office, which I haven't been to now for over a year. But it was a reminder after we solved the C subunit structure. Now we needed to do the holoenzyme. And so how is PKA regulated? So PKA and PKC both are assembled as a fully active enzyme, and then they're packaged as in a form that can be then activated by second messengers. And Earl Sutherland discovered cyclic AMP in the 1950s as a second messenger that is activated in response to neurotransmitters and hormones, and G proteins weren't discovered until much later. But now the adenylate cyclase creates cyclic AMP, and that is a major target in cells. EPAC is another target, but its major one is PKA. And that unleashes the catalytic activity of PKA. And it's not just thinking about the holoenzyme, but it's thinking about it's targeted to places in cells mainly by A kinase anchoring proteins. And so if we think about PKA signaling, we can learn a whole lot from the free C subunit and from the regulatory subunit by themselves, but really in the cells, it's the holoenzyme, and the R subunit is a dimer, and then it gets targeted to ACAPs. But there are 4 functionally non-redundant isoforms. So again, this point that the isoforms are really important and they're different. And we know from genetics that R subunits are functionally non-redundant. So, and then we have the ACAPs. So they build what is like a macromolecular signaling complex. That's what exists in the cell. And I think of this is PKA signaling islands. And you can create like several PKA signaling islands within the same cell at the plasma membrane, at the mitochondria. So it's a really important concept that one needs to really look at the larger complex and one needs to look at whole cells. And so I'll tell you just R1 alpha and R1 beta. They're the most similar in their sequence, but you can see their quaternary structures are really quite different. And their defects, they are functionally non-redundant. When you knock them out, this is embryonically lethal R1 alpha, whereas R1 beta is a depressed LTP. It gives memory defects. So if we look at the disease mutations that are associated with PKA, most of them are actually in R1 alpha, and they're scattered all over. It's a major sensor of stress in cells, and it's also the tissue-specific extinguisher of cyclic AMP-mediated gene expression, although no one's really ever sorted out exactly why that is. And some mutations make it a little less active, and they can cause dwarfism, and some can make it more active, and they can have gigantism, they have endocrine disorders. So it's very much associated with endocrine function. So if we look at just what do we mean by these signaling islands. So one really good example is ACAP79, which targets PKA to these L-type calcium channels. There are a lot of them going also to synapses in the neurons, many important signaling islands there that involve PKA. But you can see how it's anchored, and it brings also a phosphatase. This is a calcium activated phosphatase, and so you activate PKA, it phosphorylates the tail of the channel, that lets calcium come in, calcium turns on the phosphatase, and that turns, so it's a cycle, it turns it all off and on. And, you know, so if you look at going from the small proteins, the single proteins, the complexes, then how do they create these islands in cells? And as I said, you can have several signaling islands, and you can have a lot of these proteins that are part of that island are also PKA substrates. So you have little islands functioning independently in the cell. I don't think that they exchange, that they can function independently. And in addition, you can have a mechanism for taking cyclic AMP to the nucleus and mediating gene transcription there. But this is an important concept, and it's also, you know, why we need this interdisciplinary approach, that each reveals new levels of understanding about the biological function. And I think it's going from atoms and molecules to cells and tissues and then to animals. You need all of those. And my focus has been more on the structure and function, but you can't do that without understanding how it works in tissues and animals. And so that's where this working with you is so important, because you have the tools that are allowing us to understand why are these proteins so important, and why, when you just tweak it a little bit, does memory fail, aging, what happens in aging. These are really important concepts. And so this is my last PKA thing that I'm going to tell you about. Again, these are 3 different holoenzymes. You can see, even though the sequence-wise they're all really similar, you can see that the structures are really, the quaternary structures, when you look at the tetramer, are really very different. And the phenotypes are very different. So I told you about R1 alpha, but I'm going to tell you now about R1 beta, because these are R1 alpha, R2 alpha, C alpha, they're all expressed constitutively in every cell. But the R1 beta and the R2 beta are more expressed in neuronal tissues. They're highly expressed in neuronal tissues. And so we asked this question, where are they located? And so we did this study several years ago. It was published in eLife in 2017 with Ronit Elus, who's now has her own lab in Israel, and Bardo Levram. And we used our specific antibodies for R1 beta and R2 beta to sort of do a Google map. And we did the whole brain, and then we can Google down and look at specific regions. And so if we look at now, being able to now look at the whole brain, but then zoom in on specific regions. And this is what we want to do now for an Alzheimer's disease brain, and look at some of the mutants, some of the models that you have, to really look at some of the C subunit isoforms that we think are going to be really important in brain function. So you could see R1 beta, which is important for learning. It's very enriched in the hippocampus. But then you can zoom down. You could see these little green dots where R2 beta is enriched. That's Golgi. And so you can look at each cell. You know, how are the isoforms differentially expressed, and where are they particularly enriched? And it's just a really powerful way, I think, to look at PKA signaling in a broad way, and then to zoom in on the particular cell that you want to look at. So now I'm going to just focus on this new sort of discovery or unveiling. I think they had a program called Illuminating the Druggable Genome that is across all the institutes at NIH. And they had kinases and GPCRs and ion channels there. And they asked the question, you know, if we look at, these are all kinase structures, there's lots of them across the whole kinome tree, but most of them are in like BRAF and SRC and EGF receptor. And there's a lot of them where we have many, many structures. And so that's the illuminated kinome. But the druggable kinome, the dark kinome, is the one where there are really important kinases that we know almost nothing about. And that's what this program was targeting. And so within the family of kinases that were in the dark kinome is this little PKC beta. And I mean, it's shocking, you know, because PKA is sort of the poster child of the kinome tree. Everything we know about PKA, though, is PKC alpha. Everything I told you, everything everybody knows about PKA is PKC alpha. And what is this PKC beta? And so we began through this program to really look at PKC beta. And I'm going to tell you about that and why I think it's so important for neuronal diseases, and I think in particular for Alzheimer's disease. So here's the embryonic hybridization of these different isoforms. And you can see these alpha ones are all constitutively expressed in every cell. But you can see how the beta, R1 beta, I told you, is really enriched in neuronal tissues and in the brain. R2 beta is really enriched in the brain. We need to find out what these are doing there. These ones, what are they doing there now that we have our Google map? But then also look at C beta. And C beta is kind of the forgotten isoform. This was 1989 when these beta forms were first cloned. This is expression. This is RNA hybridization. It's not the protein. And so almost nothing has been done on the protein since then. And so I really do think that this is kind of a new frontier for PKA signaling, and especially in the brain where the C beta is so enriched. And so the questions are C alpha and C beta functionally non-redundant? Are they biochemically different? Are they targeted differently? And is C beta relevant for disease? And so I'll answer some of these things for you, but we're still elucidating the functional differences. And I really would like to understand how mutations in C beta can affect neurodegenerative diseases. And so I'll tell you why I think it's a high likelihood that they will. And so this disease one first. So this was a collaboration that began in 2018, but sort of the end of 2018. And my last trip to Europe was in November of 2019. And I went to Madrid to meet Victor, who had brought together this team of people. There were these patients who had these mutations that gave like a neurodegenerative phenotype, like a sonic hedgehog phenotype. There were patients in Australia, the patients in Spain, patients in France, and the clinicians had brought these patients to Victor's attention. How do they, what's the commonality of these patients? And so this was the paper that we published in 2020. And this was amazing, bringing this big group of clinicians and basic science people together. And these were the mutations. So there were 3 patients that had the same, different families that had the same mutation in C alpha. But what was really striking, there were 4 different mutations that were in C beta. And they were in different parts of the protein. And so they gave this is a very severe phenotype. It's a lot of congenital anomalies, but you could see like some of them, the extra digits. That's a sonic hedgehog kind of phenotype where you have extra digits like that. And so if you look at the different phenotypes that are associated with these patients, they have septal defects in the heart, there's skeletal abnormalities, cognitive defects, neurodevelopment defects, and some of them, more severe ones, develop some unusual tumors. But these inhibit sonic hedgehog signaling, and sonic hedgehog signaling is a big neurodevelopmental phenotype that is, PKA has really been involved with sonic hedgehog signaling for a long time, since the early Drosophila studies first unraveled sonic hedgehog. But PKA is going to be an integral part of sonic hedgehog signaling. And so I think it's all bringing it back to what is PKA actually doing in the brain. And so I'm going to tell you now some of the differences between C alpha and C beta. And so, you think, and we, along with others, there's only 25 amino acid differences between C alpha and C beta. And we thought, that isn't going to make that much difference. But you can see they're kind of clustered in these tails that I told you about. So half of them are in those tails. And so are they functionally different? What is important about them? And so here's, if we look at those C beta mutations, and C alpha 1 and C beta 1 are exactly the same size, and they differ by just these 25 amino acids. And it's the first exon, only the first exon that's different. The rest, there are 11 exons altogether. The rest are all the same. So it's only this first exon that's different. And this exon, here's this N-terminal tail. So there's a very strong helix here. This little exon here, this is exon 1. And so these, if you look at the other ones, C beta 1 and C alpha 1 are very similar, but C beta 2 has much longer. It's like over 50 residues longer. C beta 3, C beta 4, they're very short. And then these C beta 3 and C beta 4, they also have splice variants. So there's like 3 variants of C beta and 3 variants, C beta 4 and 3 of C beta 3. So there's subtle differences. It's all in this little exon, 14 amino acids at the very beginning. And so why is this so important? And this is just a movie showing, you can see the mutations, how they cluster a lot in the end lobe and in the C-terminal tail. They cluster in those tails. And so none of these are mutations that would have, the active site is right here. They're not going to affect the active site. The mechanism is going to be completely the same. They're going to be regulatory differences, I think, between C alpha and C beta. And again, you can kind of look at how these tails wrap around the kinase core, what I was talking about in the beginning, and all these mutations that are so clustered in these tails. What are they doing, and why are they so important? And which ones are most important in driving some of these diseases? And so this part is conserving the end lobe, and this part down here is variable. I told you this was conserved. This is conserved in the whole AGC subfamily, but this part is really quite variable, this part here. And so it's kind of an unexplored area now. Why is this N-terminal region so important? And again, this is the C-terminal tail. It's conserved in C alpha and C beta. There's some differences here. Those blue dots are differences in C beta, but the whole shape of the tail is conserved, and they're the same. And there's a variable region. This part is anchored stably to the end lobe, and this is anchored stably to the C lobe. And this, if there's no ATP, this is quite exposed. So the whole active site is exposed. So I think it's now, if we go back and look at the end lobe, this is now this little bit. This is the first part, this little exon 1. That's the part that's variable in C alpha and C beta and all of these different isoforms. So you have at least 7 or 8, maybe more, specific isoforms that I think are functionally non-redundant and will be really important in understanding what does C beta do in PKA signaling in the brain. And exon 1 in C alpha 1 and C beta 1 is it's a mercylated site. So there's a little ACL pocket where the mercyl group binds. And then when you make a holoenzyme, that little piece becomes very flexible, and then that helps to anchor the R2 holoenzymes to membranes. And so I think that this N-terminal part is going to be very, very important for targeting PKA and PKA holoenzymes to different organelles in the cell. And what about tissue expression? And so this, again, I think is where we can see why this C alpha and C beta is going to be so important for neuronal functions. And like I told you, our R1 alpha and C alpha are expressed highly across the spectrum in all cells. They're really highly expressed. And R1 alpha is going to be really important signal for stress, any kind of stress. C alpha is going to be, or R1 alpha is going to be involved. But they're highly expressed in all tissues. But now, if we look at C beta is expressed at low levels in most tissues. C beta 1 is expressed in most tissues. But look at how enriched C beta is in the brain. And this is just looking, again, this is by RNA sequencing, RNA hybridization. This is looking at expression of the transcript. It's not looking at the protein. And I think it's important to point out, nobody has really looked carefully at the protein, or even not carefully. People have ignored the C beta protein for all this time, since 1989 when it was first cloned. Very, very, very little is known about these proteins. And so let's look at the brain now. Again, from the early work of Stan McKnight, of looking at the hybridization in the brain of C beta 1, C beta 4, C beta 3. And you can see from this is not the protein. This is where the RNA is expressed. And you can see C beta 1 is highly expressed in the hippocampus here, which is where we found R1 beta was highly expressed. So it's got a very distinct expression pattern. But if you look at C beta 4, its pattern of expression is very different. And C beta 3 is expressed at lower levels, but it's also expressed in a very specific way. So I think it's this expression. How can we now map this at the protein level? So, which is one of the things that we are really hoping that we can do. And then if you look now again at specific cells, you can see how tissue specific these are, these isoforms. So C beta 2, that's that very long one. It's not expressed in the brain hardly at all, but it's expressed in lymphatic tissues. So it's expressed in lymphocytes. So it's highly expressed in lymphocytes, and it's actually upregulated in cancer. So C beta 2, I think, is going to be a real marker for inflammation when lymphocytes invade any tissue, maybe even in the early stages of Alzheimer's disease. C beta 2 is a marker. It's going to be a marker of lymphatic tissues. And whereas look at C beta 4, you can see how specific it is for the brain. And C beta 3 is also really specific for the brain. So how can we look at this now in terms of mapping the protein? Can we do that kind of a mosaic imaging of the brain for these beta isoforms? And I'm going to tell you what we've been doing. And we have now samples from Robert for human Alzheimer's disease brains. And we also have samples of the mouse models, the A beta mouse model and the tau model for Alzheimer's disease. And one of our goals now is to really map those. But we've used the retina as kind of a window into the brain. And I hope you will agree that this is giving us a really good insight now to go back to the brain to use these same strategies for the brain. But so this has been, Dorota has been a remarkable colleague. She was here, had been a former postdoctoral fellow with Jeff Rosenfeld. She's really interested in gene transcription and in the retina. And she's now up at Irvine in their ophthalmology department, which is also a really rich source of an interdisciplinary approach for studying the retina. And they also have an ADRC at Irvine. And so I'm hoping our two can communicate together. And this is from my lab. Frank Ma has been doing the imaging here, and Janae Rao. They'll be the ones that will do the imaging of your samples as well. And then Zbigniew at La Jolla Allergy and Immunology, and Lan, who is a postdoctoral fellow or research assistant at Irvine with Dorota, have been doing these studies. And so if we look at the retina, and I'm learning, and I hope you appreciate that. I'm really learning a lot, even now at this late stage of my career. This has been an incredible time for mentoring me about neurons. And although Palmer knows all about the brain and synapses and things, he doesn't talk about those things very much to me. So I'm really learning a lot about neurons and neuronal function from this collaboration. And so you have the cells. The retinal ganglia cells are the ones that are myelated and go way back to the brain. And so most attention is focused on the retinal ganglia cells. But you have these interneuron cells here, and then you have the photoreceptor cells. And so these are highly differentiated cells in the retina. And we use this as a model system to look at not only the art subunits, but specifically the C alpha and the C beta. And we asked, are C alpha and C beta localized differently? Are they functioning differently? And so here's the imaging that we get for the C alpha. And here's the PKC alpha. This is PKC alpha. It's a good map of synapses. So it's a really nice control for synapses in these regions here. And you can see that's rich in synapses here and rich in synapses down there. So this is PKC alpha, and there's a lot of them up here in the photoreceptor cell region, and it's in all the layers. But this is PKC beta. PKC beta is in the it's in these synapse regions as well. It's particularly enriched in some of the retinal ganglia cells. It's also up here in the rod cells and the cone cells. And so you see some very specific regions that are labeled differently by these C alpha and C beta. And I want to tell you what these regions are, to look at those more closely, so that we can begin to understand, are these really functionally different? Are they localized differently? And yes, these studies would tell us they do seem to be really localized differently. And so here's the C alpha and the C beta expression. And I'm going to go down now, look really closely at this region here, more closely. And so what are these regions down here? So these regions down here are what they call the cilia transition zone. So this is a transition zone between the outer segment and the inner segment, and that's a cilia transition zone. It's a very focusing, and it localizes, organizes transport into the cilia and out of the cilia. What are these up here? This is the outer segment, and this is interesting also because this is C alpha, it's the only one. We've looked here at C alpha, C beta, R2 alpha, and R2 beta. And C alpha is the only one that's in the outer segment at all. The others aren't there at all. And what's also interesting about this is that C alpha is on the plasma membrane. It's not in the discs where the rhodopsin is, it's on the plasma membrane. And I think that's also interesting for this sonic hedgehog signaling. But most interest is what are these things that are being labeled by C beta, these ellipsoid, large ellipsoid, they're not vesicles, they're not membrane bound, but the compartments. And you can see this is a cone cell, this is a rod cell. So you have compartments within the rod cells too, but the cone cells are just striking. And so this is just showing you with the R2 alpha is R2 beta is similar, C alpha is similar to this. And that what I just want to emphasize here is first that the red part is the cell body. R2 alpha is all throughout the cell body. It's not in this ellipsoid. C alpha is all throughout the cell body. It's not in the ellipsoid, but PKC beta is the one that is specifically in this ellipsoid. And this ellipsoid is known to be mitochondria. And so we asked to confirm or validate that this was indeed mitochondria. We used a marker for oxidative phosphorylation. It was I think one of the subunits of cytochrome C4 oxidase. That's a marker for oxidative phosphorylation. And you can see how C beta and OXPHOS really go together across these layers here. And this is looking at now a little bit closer here, looking at that co-localization of PKC beta with oxidative phosphorylation markers. And even in the retinal ganglia cells, you can see that they do co-localize with the mitochondria in these regions here. So then we look more closely at this, and this is just a close-up because I think, to me, this is like structure at just a different level, but this is looking at the localization now of an oxidative phosphorylation marker with C beta, and showing how beautifully they overlay. And with our colleague at La Jolla Allergy and Immunology, we really went through and did all the Z-stack layers of the mitochondria. And I don't know, and it's a really important question now to resolve. Is it inside the matrix of the mitochondria? Is it on the inner membrane of the mitochondria? Is it on the outer membrane of the mitochondria? Those are really important next steps to answer. But it's really enriched throughout the mitochondria, and we have these really remarkable images that show C beta, I think, is functionally distinct from C alpha, and it's going to be doing some important things. We don't know what those things are yet, but I think in terms of organelle dynamics, it's going to be really important. And so my idea is that we can use this as, what happens in light and dark. Those are things we can image for. What happens in fasting and feeding, what happens in aging, diabetes, and also Parkinson's disease and Alzheimer's disease. What are the differences that we can see if we look? These are all human brains. This is all human brain that we've been mapping in the retina. So I think it's going to be kind of a marker for dysfunctional PKA signaling, I think, but for a lot of diseases. And I'm convinced even night and day and fasting and feeding, you will see differences in these mitochondrial regions. And so I think, can we use PKA as in the retina as a window into Alzheimer's disease? That's my hypothesis, and I hope that we will be able together to figure out what PKA is doing in the brain and what is it doing in Alzheimer's disease. And is mitochondrial dysfunction a common theme of neurodegenerative diseases, Alzheimer's disease, Parkinson's disease? They all ultimately lead to mitochondrial dysfunction. And can we use that? Can we do live cell imaging of mitochondria in retinoids, organoids of the retina, and organoids of the brain? With our Parkinson's disease mutants, we're poised to really look at the mutants in brain organoids. But I think we can use the retina organoids in the same kind of way and just look at actually an organoid that you can make from a patient's, you can make iPS cells from a patient, and you can look at that particular phenotype, express it, and look at mitochondrial dynamics. That's kind of a global vision. Let's see, oops. Need to move on again. Take this away. So I have some references that are there's a lot of references in the literature that say PKA phosphorylation is really associated with neurodegeneration and Alzheimer's disease. And this is tau phosphorylation is a big factor that's associated with PKA being one of the major kinases that can phosphorylate tau. Two other papers. This is the exome sequencing reveals the PDE, phosphodiesterase, as a candidate for early onset Alzheimer's disease. Tau inhibits PKA. So what is going on? This is a PKA R2 holoenzyme. What's the crosstalk that's going on between PKA and some of these neurodegenerative phenotypes at synapses? And this is another paper that was a couple of years ago, well, that's a couple of years ago, 2019, a microRNA that actually regulates the PKA C-beta gene, as well as one of the secretases that's involved in the A-beta processing. So there's lots of clues that say PKA is going to be playing a major role. And I wanted to go through this. I hope it was not too disconnected, but these themes, because I really think we need you to really understand the translational consequences of these mutations. And can we use this new discovery of C-beta as an important regulator in the brain as not only a way to look at PKA signaling in the brain, the normal biology, but also to look at disease. And I think it's a kind of opportunity that we can build on the kind of expertise I have, but we need the kind of expertise that you have. And so that's my message to you. And also, you know, I've acknowledged the people who have really contributed, and especially I want to acknowledge Dorota, who's been a more recent colleague this last year, and Janae and Frank, they've been doing a lot of the imaging, and they're the ones who will be working on the samples that we have from you. So I think it's just a wonderful opportunity to leverage our collective expertise and see, can we really make some major inroads? And I want to end by acknowledging Roger, who really used chemistry to understand biology and, you know, did all this work here. He was such a mentor for all of us. I think now sequence, structure, function, that's my mantra. And I think that's a window into biology, but now it's a window into disease. And so I think the disease mutations, like I said, are kind of a treasure chest, but Roger has been an enormous influence on me, as he has been on most of you. And Roger would always say, it's not enough to have a structure, Susan. You need to understand what those molecules are doing in the cell. And of course, he was always right. I probably wouldn't have done many of these things had it not been for Roger always there saying, you have to understand what it's doing in the cell. So with our kinase expertise, Alexandra and me, with Jean Zhang, who's also doing all this super imaging of PKA in cells, and all the clinical expertise that we have here, especially in Alzheimer's disease, I'd really like to use what resources I can in my lab to help us understand this. And so thank you so much for inviting me. And I hope that I can engage even more of you working with Robert, but even more of you possibly as collaborators. So thank you so much for your attention.